Hello, everyone. I hope you had a really good weekend, and I hope that you are going to take some time to enjoy yourself over Thanksgiving as well. This will be a pretty straightforward lecture on the basics of the skeletal system. We will talk more about some of the more pressing details, calcium, things like that, when we meet together for lecture next a week from Monday. So I'm just going to go through this, and hopefully you follow along. Know that you can speed up my voice because I try to talk pretty slow, especially while I'm writing, and you can do that just using YouTube's speed up feature. You can also get a copy of the notes on Canvas as well, so I'll upload those just after I upload the video, so you should have that available to you. There is a case study associated with some of this that should be up shortly. It won't be due until after you get back from Thanksgiving, so no rush on it, but do make sure that you're sort of keeping up so that the last couple weeks of the quarter go pretty well for you. Okay, so I'm going to go ahead and get started, and hope this goes well. All right, so first of all, we have to talk about the skeletal system in terms of how it functions as an organ system. And that means we have to remember that organ systems are organs that are working together for a common function. Now, the organs in the skeletal system are not, sorry about that, are not very complicated. We have our bones, of which there are approximately 206 bones in adults. There are about 270 as infants, but then some of them fuse, so we call them all one. Plus all of your other cartilage and connective tissue. And so those ligaments, tendons, those sorts of things work together in terms of joints, and then all of these sort of pieces work together for a few different common functions. I'll list those over here. So the first one, most important one, is support and protection. Right? We all know that our bones are here to hold us up, keep our organs safe. All of those soft sections need lots of protection. But they also have a function of being the site of muscle attachment, where without bones, then muscles wouldn't have much to pull on. Remember, we talked about muscle action, and so muscles have to pull on an origin or on an insertion, move it towards an origin. That's the only way that they can move, and those bones provide the attachment for those. Bones also provide us with calcium storage. You know, most of the time when you hear about calcium in terms of nutrition, you hear calcium makes bones healthy. But really, one of the things that we have to think about is what we already know about calcium, which is that calcium helps us to release neurotransmitters, and that helps us to send nervous signals. So that's a really, really important function that is kind of outside of the fact that calcium makes bones hard. So, but we'll still write that down. Calcium makes bone hard, so it can be a better protective feature, but it can be pulled out of the bones, and we'll see how that works shortly. So that's what I mean by storage, that we can pull that calcium out of our bones if we don't have enough from our diet. And then it can help release neurotransmitters. So for exocytosis, and it also helps us with muscle contraction. Calcium is a pretty important part of that. So calcium homeostasis, really, really, really important. Okay. Another one is what we call hematopoiesis, which is the formation of blood. Hematopoiesis. You know that the bone marrow from our lab is found in the bones, in that medullary cavity, and so this is how we make our blood cells. Location of making the blood cells. You'll learn more about that when you do your cardiac unit in MP2. And then, last but not least, it's also a good place for lipid storage in the yellow bone marrow. Okay, so the red bone marrow is mostly where we're doing blood formation. Yellow bone marrow is made up mostly of lipids. We saw that in lab as well. Okay. When we break it down by tissues, just to make sure that we have a really nice list, we have all three types of cartilage, and we also have compact bone. We have spongy bone. We have adipose, which is a connective tissue. So is all of these other ones as well, but it's easy to forget that adipose is connective. And then we have dense regular, CT, which makes up the periosteum and the endosteum, and then is continuous with our ligaments. Okay, so let's make that a decent size. So support protection, site of muscle attachment, calcium storage, hematopoiesis, and lipid storage. Okay, so really important in terms of our functions, and then we have this full list of tissues that we're going to be sort of using in different parts. Now, we already saw long bone in our lab, and we identified that it has these sort of areas that we focus on, the diaphysis, which is the center of the bone, and then there's either a proximal or a distal epiphysis. So on this one, this will be the proximal side, and this is the distal. This is one of the bones that's found in your lower leg. Okay. On this diagram, we have right in here, so all of this blue shaded area, that is where we find our compact bone. Okay. And that compact bone is surrounded by an outer layer of connective tissue, which is the periosteum. And on the other side, so moving all the way to the inside, to right on the inside of the medullary cavity is the endosteum. So then this cavity is the medullary. Okay. Both the top and the bottom of this bone, distal and proximal sides, can be spongy bone or have some spongy bone, depending on the particular bone. And then the entire top part is covered in articular cartilage. So that would be this area up here, but then also anywhere else where there's an articulation, we would see that articular cartilage as well. Okay. Now, those are the long bones, and that's mostly what we saw when we were in our lab, but we also have a few other types of bones. We have flat bones, which are the bones that make up the skull. We have short bones, which are the bones like the carpals and the tarsals, which are our wrist and ankle bones. We have irregular bones, which are things like the vertebrae. And then we have what are called sesamoid bones. So these are the little bones in joints. So an example of that would be the patella, which is the little bone we call our kneecap. Okay? So flat bones, short bones, irregular bones, sesamoid bones, and then our long bones, which are the ones we focus on most when it comes to growth. Okay. So then let's look at a little bit more anatomy. We're going to look at how the tissues and the cells are functioning bones. Let me just double check that we're still recording well. Hold on a second. Okay, looking good so far. All right, so this is a close-up of what we saw already when we looked under the microscope, where, let me see what I can highlight. So this whole structure that's all in yellow right there, that whole thing would be an osteon. And then within that osteon, we have those concentric lamella that are surrounding a blood vessel. Which is in a central canal. So that can give us some nourishment. What that blood vessel in the central canal means that this is vascular tissue. It has vessels, blood vessels, that can help us to grow. So this is what we've seen in the lab, compact bone. But another structure that we didn't look at under the microscope, or another type of bone tissue, is that spongy bone. And the spongy bone, while it has a similar sort of structure to or sort of organizational structure to our compact bone, it's not exactly the same. So this is a section of spongy bone, and then down here we have this sort of zoomed-in view of one part of the spongy bone. That little section is called a trabecula. Trabecula. That's T-R-A-B-E-C-U-L-A, trabecula. So that's just a zoomed-in view of one of those. And one of the things that's pretty obvious when we're looking at spongy bone is that it's full of spaces, right? As opposed to compact bone that's very compact, spongy bone looks like a kitchen sponge. And the reason for that is that the spaces are filled with bone marrow. So they have their own, or their function has to do with moving around bone marrow, needing a lot of blood vessels for growth. So this is where we find bone marrow, or one of the places that we find bone marrow. And then instead of having a central canal, they, inside of those spaces, oh, no, goodness, let's see, where's my, so no central canal. So instead of having a central canal, the blood vessels are in the spaces as well. All right, so going to the zoomed-in view, this trabecula, this otherwise has these lamella that look very similar, and then the cells that it has are also called osteocytes in lacuna. So there's not much of a difference in terms of the basic structure. You have these osteons sort of things in compact bone that are exactly the same as trabecula. In spongy bone, the biggest difference being that the blood vessels are on the outside versus the inside. Okay. We also, our osteocytes can be called clasts and blasts, if you remember those terms from way back when we talked about connective tissues, where we're going to get into a little detail about what those things do in just a couple minutes here. Okay, so cells, osteoblasts, osteoclasts, osteocytes, we also have, because this is connective tissue, fibers, right? So we have fibers and we have a ground substance because all connective tissue does. And the fibers plus the ground substance together make up what we call the matrix of this tissue, which is a solid matrix. So the fibers that we find in bone are collagen, and this makes bone flexible. We don't think of bone as being flexible, but would break far more often if it wasn't at least a little bit flexible. And then our ground substance is solid, and it's made up of a material that's called hydroxyapatite. Hydroxyapatite. And this is a calcium plus phosphate, substance. It's very strong and very durable. Right? So this hydroxyapatite is the sort of making of the hard part of our bones, but it combines with the fibers to be a little bit flexible. Okay? So when we see a section, in particular of compact bone, because that's really what we're going to focus on, we know that we have this dense, regular connective tissue on the outside, right? And that helps us to protect the structures inside. So both the periosteum and the endosteum are made of that dense, regular connective tissue. What we haven't talked about is that right underneath that connective tissue is where we find most of our cells in fully formed bones. Okay? So those cells that we're looking at, those are the osteoclasts and osteoblasts. They don't look functionally very different, but we know that they do very, very different things, where a blast builds bone, and it does so by secreting that hydroxyapatite. And collagen. Or we could break up bone using osteoclasts. Okay, so clasts destroy or break apart bone, and then osteoblasts build or make bone. Osteoclasts, when they're functioning to break apart bone, they use an acid. I'll talk about it in a little bit. Okay. Now, this process of breaking apart bone and making bone, we do this all the time. It's not something that we do just on occasion. We do this regularly. And the main reason that we're doing it regularly has to do with hormone levels. Let's say BC, because hormone levels and physical activity. So doing more high-impact physical activity means that we're building more bone. Doing less physical activity, we lose some bone. It's one of the issues that they have with space travel, because there's not enough gravity on the bones. The bones get pretty weak in a pretty short amount of time. Okay, so periosteum and endosteum, that's where we find most of the cells. And then within those osteons, not as many cells, and most of them are not functional anymore. Same thing goes for the trabecula. We're mostly seeing our osteoclasts and blasts on the outside of the trabecula. All right, so now we're going to look at two different types of growth, and then that's pretty much going to be all we're working on today. So the first kind of growth, this growth is called intramembranous, where we know that intra means within. So this is within a membrane, and then ossification is to make bone. And this type of bone, intramembranous ossification, is primarily flat bones that are using this. So the cranial vault, so all the bones of the skull, and then the facial bones as well, use this kind of growth pattern. Okay. So the way that we do this growth pattern, I'm going to get really big here, so leave yourself some room for notes. So the way that we do this is starting off over here, so this would be the first step. We have these mesenchymal cells. Okay, so mesenchymal cells. Our mesenchymal cells are what we call undifferentiated. Do you remember that term differentiation from way, way back in your 160 material? Differentiation is where a cell gets a job, essentially. So undifferentiated, embryonic connective tissue cells okay, so they don't have a job yet, but they will. Okay, and then over time, so this arrow is telling us that time has passed, those cells become osteoblasts. So mesenchymal cells become osteoblasts. Okay. When mesenchymal cells become osteoblasts, then that means that over time, those osteoblasts begin to secrete collagen and hydroxyapatite, which is what makes up our bone matrix. Okay. Any of our osteoblasts that get surrounded by the matrix become osteocytes, and they can stay as osteocytes as long as they have some form of blood that can reach them, so vessels that can reach them. Otherwise, they die off eventually. Okay. So essentially what we have here, we have these mesenchymal cells that start off. They become osteoblasts. Osteoblasts then start to secrete, and they're secreting all of their hydroxyapatite and collagen matrix towards the inside. Osteoblasts sometimes get surrounded and become osteocytes, but over time we have this bone that's developed between this membrane that's on either side, so we call it intramembranous. So this is a pretty basic, pretty simple, straightforward way of growing bone. The places where we see this after birth are primarily in the fontanelle, which we saw in the lab. So this is an area where infant skull growth can occur. Using that intramembranous ossification. And then we also see them in the sutures. So at the sutures, growth can occur. Okay, and they're connected by a very fibrous connective tissue. So it's dense, regular connective tissue, but extra, extra strong. So at the sutures of the skull, we can have some growth when we're young. After a while, those sort of fuse together and become a really strong barrier, protective for the brain. All right. So our next type of growth is called endochondrial ossification. And this is, so chondrial has to do with chondrocytes, endo inside, so chondrocytes that grow from the inside of our bones. So this is the growth of long bones. Okay, the growth of long bones. So we have the same mesenchymal cells that start these things off. So those are those undifferentiated mesenchyme or mesenchymal cells. Um, so in this process, rather than mesenchymal cells becoming bone cells directly, the mesenchymal cells will become chondrocytes. So our mesenchymal cells become chondrocytes, and those chondrocytes will start secreting cartilage matrix. When they start secreting that cartilage matrix, they make what they call a cartilage model. So it's a little model bone, right? And that model bone is, clearly before birth, and it's a very flexible bone, so it's clearly not enough, right? So then the next step that happens is we start to form this bone collar. So this is where we start to actually have some cells that have turned into osteocytes. And eventually that bone collar will hollow out the inside, which is that medullary cavity. Okay, so that starts to form pretty soon after we have that bone collar model, and then over time the chondrocytes turn into osteocytes, form more and more compact bone around the edges of this space, as well as here. And then where we really start to see bone growth as adults is on either side of those epiphyseal plates or growth plates, as this says, where they get larger and then they fuse as adults. Okay? So what we really have to think about is what's going on in this space right here. And then it's also happening in this entire space in the early stages of development. Okay? So what we care about, again, is our epiphyseal plate, which is the site of growth in long bones. Okay. And it is, because when I say the site of growth, that also means the site of mitosis, so lots of mitosis. And then it fuses at puberty. Okay. For females in particular, males, it normally fuses about 2 to 3 years after puberty. Okay. So let's talk about what or how that happens on a cellular level, because we know we love cells in this physiology class, right? So when I said that we may have these mesenchymal cells that become a chondrocyte, that's what's going on here. So down at the bottom, we've got all of this cartilage matrix that's filling in this space, right? And then inside of that cartilage matrix, we have these chondrocytes. Okay. So this is our matrix, and this is a chondrocyte. Okay. So after the chondrocytes have made all of that matrix, then the matrix will start to calcify. As in, we add calcium to that matrix. Oh, that should be IES. Okay, so the matrix calcifies. When it calcifies, this kills the chondrocytes because they no longer can get the nutrients that they need through the matrix because it's too hard. Okay. So here in this space, so we have our chondrocytes here. These are still chondrocytes. So these are dead chondrocytes. Okay, and those dead chondrocytes will start to be replaced in this last section with osteoblasts and osteoclasts. Okay, so these are the dead chondrocytes, and or calcified cartilage with chondrocytes that have been already removed, I guess, where our osteoblasts will move into this area and they'll start secreting bone. And the osteoclasts, at the same time, will be remodeling the bone and cartilage so that it's in this right location. So that's where we have that bone color start forming, right? So the osteoclasts are coming in, they're taking out cartilage, they're removing little bits of extra stuff in there, and then they're replacing it, the osteoblasts, with bone in the right locations. Okay, and then again, like we have one over here, they become osteoclasts when they are trapped. Okay. So when we're looking at this idea of growth, we have our cells that start off as mesenchymal, turn into chondrocytes. From chondrocytes, they turn into osteoblasts and clasts, and then osteoblasts and clasts live in that tissue throughout the rest of that, the lifetime of that human, right? Still making bone and breaking bone as needed in order to have access to cartilage or to be stronger because we're being exposed to more exercise. Okay. So one of the things that we know is that these are really affected by growth hormones and also sex hormones, and this is because our growth hormones, GH, cause our chondrocytes to divide. So they act as a signal for those effectors, and they stimulate the chondrocytes to divide. So do some mitosis, and also to secrete matrix. So that can cause the epithelial plate to widen. So that's one of the things that growth hormone does, because once that epiphyseal plate widens, then the bone lengthens. Okay, and then when we have sex hormones, when we're thinking about sex hormones, then the main function that sex hormones are going to do is they're going to stimulate osteoblasts, and they're also going to stimulate growth hormone. So you have kind of a double whammy, though. In particular, that's true when we're talking about testosterone. And after we age, those sex hormones and growth hormones sort of change in their amounts, and so that slows and stops growth eventually. Okay, so what happens at that point is we have this at the epiphyseal plate, we have the bone overgrow the cartilage, and that's mostly because the osteoblasts make the bone grow, and then that turns that epiphyseal plate into a line, and then growth stops, at least growth in terms of height and length of bones, or length of long bones. All right, so that's all we're going to do for this lecture. I'm going to leave it at that, and then we'll do the rest of this on Monday when we come back, and we'll also talk a little bit more about calcium because we're getting pretty close to our endocrine section. So we're going to be finishing up a little bit of skeletal, moving on to muscular after Thanksgiving. And we'll see you all on Tuesday for your lab, where we will be discussing the arm, so that'll be exciting. And until then, I will wish you a good day. Okay, bye-bye.